All right. Well, good evening, and thank you all for coming here. This is part one of a series of webinars slash workshops that we're going to be doing. And today, what we're going to be doing is just kicking off this whole series here. The series starts with this webinar today. Today, we're going to be discussing a few things. I will get into those details soon, but also this will be part one of three. So we'll have this webinar and then two workshops, which would be in person. These workshops will be hosted at Dan Kittredge's farm of the Bionutrient Food Association. And again, we'll give more details on that. So the purpose of this particular webinar is to essentially explore the microbial world and the soils and significance in agriculture. So let's go ahead and start up right away. Here we go. Just a few things, housekeeping. This webinar, as you all know, is being recorded. We will be pausing a few times just to field any questions. If you have any questions, please put them in the chat. We will be pausing throughout the webinar to address some of these questions. And in addition to that, we will actually have some interactive polls and quizzes throughout too. So please don't fall asleep. Again, so the purpose of this event is to actually create some type of understanding behind the microbial world and the role it plays in agriculture. We will also be discussing some tools, the tools that we actually use to interact with these microbes in terms of quantifying and just, really monitoring. And we'll be touching upon two particular tools. This is going to be a microscope. We'll go over some of the microscope parts. Hopefully it's not too boring and dry. And also the microbiometer. The microbiometer is a tool which, again, we'll get into more details about that. But those are the major tools that we're actually using to identify these microbes. And one more thing, if you don't mind muting yourself while not speaking, that'll be great, just to not interrupt the audio. Right. All right, so let's all get started with the soil food web. And before we get started with the soil food web, I actually wanted to get us started with a poll on knowledge or prior knowledge, just to see where we are in the group. So if you can all actually just answer this one simple question here.
Hi, Ruben. I just have a question of what is the low end choice? What's the high end?
So sorry, yeah. So low end would be I know nothing. Which one would be I know nothing. Five, I'm a master. And I think we have a critical mass of that. Maybe one more person. Although I know that some people are driving, so I don't expect you to do this if you're driving. So let's go ahead and end that poll, actually. Wonderful. So we actually, let's go ahead and share the results so that you all can see. We are doing this in community. I want to make sure that um, there are no such thing as experts, in my opinion, and we're all learning collectively here. And I can see that, we're pretty much everybody's falling between it's a pretty even distribution amongst, intermediate and a high level. So this is great. Thank you. And let's see. So let's go ahead and dive right in here. So I'm sure that most of you have by now have actually seen this graphic here. And I will preface by saying that this is a very simplified view of what the soil food web is. Um, I wrote down on the side here that the soil food web is a complex network of interactions among soil microorganisms or organisms. And I want to emphasize the word complex. This is much more complex than what you see here, but let's go over some of these uh, microbes, what they call trophic levels, or you can see at the bottom here. Hopefully everybody can see on the screen here. It really all starts with our beautiful sun shining down on our plants, and from there, this entire process actually moves through. So at the first one, you actually have your photosynthesizers, which are your plants. Your second level or trophic level in your ecology here or soil ecology, you can find your decomposers, mutualists, pathogens, parasites, root feeders. All that means is it's mostly bacteria, fungi, and a few nematodes. Um, Next, we actually have our shredders, predators, and grazers. We'll explain in a little more detail, actually, what each of these do and how they contribute to this complex soil web. Uh, Next, we actually have the higher level predators, which are extremely important in the system. And again, we'll go into more detail on that. And our megafauna, or actually not megafauna, but fauna in general, birds and mammals that are actually much
American Medical
higher.
Society Foundation.
And so these are the trophic levels. These are the microorganisms that when we actually speak of this soil food web, these are the organisms that we're actually speaking of. The ones responsible for all these interactions with plants, meaning that they actually are providing nutrition, protection, and also just diversity to the soil and resilience to the soil and plants too. So let's go ahead and move on to the next slide here. All right, so this slide actually is a little bit complicated, and I want to take a little bit of time explaining what you guys are seeing here. As you saw from the previous slide, we actually looked at some of these predators. And so the predators that we're going to be speaking of in particular in this slide are protists or protozoa. When we're looking at these, and again, we're actually talking specifically about using a microscope here. When we're looking at these protists, we're actually looking at specific morphology. And when we're referring to these protists, we're looking at three groups. We're looking at flagellates, amoeba, and ciliates as well. So hopefully y'all can see my cursor here. Here's the legend that we're going to be following. On the left bottom side here, which you have your soil protists, bacteria, fungi, mycorrhizae, and roots. And on the right-hand side, you actually have a positive effect, which is delineated by the green arrow, and the negative effect by the red arrow. and then an acronym here of dissolved organic matter. So let's start up here with the A. As you all can see here, you actually have the protists, which again, green arrow indicating that you have gram-negative and gram-positive bacteria. These are actually going to be consumed, or the protists are going to consume these. By consuming them, they're actually going to release plant nutrients that are in plant available form that the plant can easily take up. That's here is labeled as the microbial loop. However, we kindly called it the poop loop, meaning that whatever these organisms excrete, they are actually going to be excreting any residuals that the plant actually can take up. And those are actually nutrients that they're providing. Next up, we actually have some, again, both positive effects here. And from you can see here, we are looking at how these protozoa actually stimulate the bacteria in particular to create these secondary metabolites. These secondary metabolites are also known as plant-promoting growth hormones, essentially chemical signals or chemicals that will help the plant either fight disease or suppress disease, and also help it grow. Next up, we actually have down here C. C are the chemical interactions. Both C and D actually are mostly chemical interactions, but for C, what we actually are seeing here are both the positive and negative effects that these protists or predators actually can have in the system. These now you can see that that there's some pathogens. Some of these protists can be pathogenic, while others can actually be of benefit to the plants. And again, D illustrates the same thing. However, it's all positive in this regard, with these protists actually being the safeguards of the plant. Next, we have E, which is the complete opposite of D here, which some of these protists may actually just be pathogenic. And if you're wondering too, if well, why would we actually want these organisms? And this goes back to the idea of actually creating diversity. We don't know exactly what each of these microorganisms do. And so promoting biodiversity is actually going to be key to protecting your plants from such pathogenicity. So you can actually see that it's well illustrated down here at D, where when you have diversity, your plants are protected. But once you actually have some type of specific microbe that is probably overrunning the system, that can have detrimental effects. And similarly as well, you can actually see on F here, where your protists are actually consuming some of these beneficial microbes as well. And we'll get into why some of these are, particularly bacteria. We'll actually get into as to the why these are actually beneficial to the plant. So let's go ahead and let's see. Let's go to the next slide. So I would be remiss if I actually didn't show you guys this. This is actually something that I haven't shown the public yet. Something that I'm really happy that I was able to capture this. I captured this image or this video a few years ago. And what you're going to see here is, and I apologize for the low quality. I didn't have the good camera that I have now, so I actually used my phone. And while I was doing this, you'll see that my daughter was actually there with me. But I kept it there because I also want to show folks that this is not complicated, and you know, even my child actually gets behind my microscope and we search for microbes, and she's well aware of them as well. So I'm just going to play this quick video so you guys can actually see this predator-prey interaction that's happening here.
Wow.
Small one, but delicious.
It's very small.
Smaller than.
Look at him. He looks big.
Looks big.
Yeah.
So what you're seeing here is a predatory nematode, and this predatory nematode is actually consuming a unknown nematode. I was not able to determine what type of nematode it's consuming, but for the most part, some of these predatory nematodes do consume more of those pathogenic ones. They do prefer to go for some of these pathogenic ones. I think the reasons are unknown. But they do tend to feed on them. And again, by this nematode consuming the smaller one, it will have to actually poop out additional nutrients, and those nutrients will be in plant available form for the plants to take. So it's actually serving a dual role, right? It's actually providing this nutrient cycling that we just talked about. But it's also providing some protection against a potential pathogen that can infect the plant roots as well. And I don't know what my was doing there. Probably cheering on the nematode. All right. Hit the red button. All right, so next up, we actually have the inverse, right? We actually have this, so A, B, and C, these are all actually a type of fungi known as nematode trapping fungi. There's a whole slew of them, very interesting. And what they do is that they actually, they're typically saprophytic, meaning that they actually they're not predatory in nature. But what happens is that once the nitrogen reserves are low, what they do is that they actually weave these hyphae around in the soil to create, the best thing that I can describe it as is perhaps a spider's web, right? And similarly to the spider's web, what the fungus does is that it allows the nematode to crawl through it, it gets stuck, and then you see these concentric rings on the right-hand side here. Once the nematode goes through there, it gets trapped, tightened up, and it's slowly consumed from the inside out. So again, this goes to speak to the diversity that's in our soil and the complex system that we also have. Another thing too that I wanted to point out was, and this goes back to the protist slide that was there, the researcher that actually conducted that research on, or created that paper, he made a comment once about, we believe that, these that there's certain interactions that the predators are actually consuming certain things, and he gave the example of a nematode that he had in the lab with a bunch of amoeba. Now, typically amoeba will go for smaller predators such as bacteria, but in this case, when left in a laboratory setting, the protists actually started consuming the nematode. So this speaks to a few things. One, we don't know anything, and two, that these are very, very complex scenarios where a lot of times we're trying to control something, and we may actually have a different outcome that we actually think. And this brings me back to a comment of a that I like to mention when I'm having these conversations from a Mexican biologist, microbiologist. He made a very good analogy of, you know, he brought in some microbes into the lab, and let's say, or actually, let's say I bring in somebody into the lab and I feed them a bunch of beans. And then I go out, write a paper saying, hey, this person actually only ate beans. They were so starving for beans, that's all they ate. But when you ask that person, they're like, no, I hate beans. It's just that that was the only thing that they fed me. So we have to be careful as to when we're looking at data coming from the labs, when we're dealing with living systems. It's very, very complex. And I'm not discounting any of this information, but it's all important to actually have that in the and take it into consideration. All right. Next up, we have the rhizophagy cycle. And we couldn't complete this soil food web talk without actually speaking on this rhizophagy cycle. Now, I'll go over this really quickly here. In 2010, the University of Queensland, there was a group of scientists there that discovered that, you know, some of these things that were perhaps thought of as chloroplasts that were inside the, or protoplasts that were inside of plants were just that, you know, nothing, just, you know, just some chloroplast. And what they found out was, in fact, that
Recording in progress.
That these were in fact bacteria. So fast forward to 2019, a group of scientists out in Rutgers University, led by Dr. James White, discovered that this was actually a cycle. And as you can see here in this picture, what they discovered is that the bacteria were actually entering from the root meristem. The plant would actually send out root exudates, you know, which are just, you know, organic acids, amino acids, all these different chemicals would be released through the roots. Once they were released, they would attract certain bacteria. And by the way, specific plants attract specific bacteria, especially depending on their needs as well. So they would attract these bacteria, they would take them into their roots, and through their roots. This process of cyclosis would start happening, but before the cyclosis, the plant would actually hit them with NOx or superoxide, and it will destroy the outer membrane of the bacteria. Once it was in there, it'll start doing this, you know, machine washing type cycle. And if that bacteria was, for example, consuming a lot of nitrogen or phosphorus, whatever it was consuming, now the plant actually has this inside. So this was actually a way where they discovered that plants get their nutrition without our intervention, as long as they actually have the right microbes there. What's amazing about this process and why it's actually a cycle, as you can see here, part of this actually, the microbes will actually select the site and start pushing. And actually, that's how we discovered that microbes are responsible for root elongation. Root elongation only happens when the microbes are present there. And so they push. The roots get elongated until they actually burst out of the roots again. Now, the beauty of this is when the plant actually says, well, wait a second, before you leave, I think that you came in here with certain parts, like, you know, maybe you had a flagellum, maybe you had other moving parts. Why don't I give you all the material that you need so that you can rebuild yourself, and let's have this party again. So really, really important to understand this. Again, this is something that's fairly new in understanding, but something that we should all have a basic understanding of. And I think I think that's the end of this, and I want to actually go ahead and put out a quiz before moving on to the next part, just to make sure that nobody fell asleep on me. And that's the Soil Food Web quiz. It's just I think it's just one question.
No, Domino's doesn't deliver here, it's Gabby's.
All right, we'll give it a few more seconds. We have about 83% participation. And the question, for those that are probably driving, the question is, which of the following organisms are part of the soil food web? Bacteria, plants, insects, or all of the above? And I think it's fair to close this now. We can share the poll. So as you can see, the answer is all of the above. All of these, if you recall the slide a few back when we were talking about the trophic levels, you saw the plants, saw the arthropods, insects, mammals, birds. So question all of the above. Thank you for participating. So I want to actually, before we move on to the next slide, I want to see if we actually have any questions. I can't see the chat, so Melissa, I don't know if you're in a spot where you can actually field any questions if they're on the chat, or just actually come off of mute if anybody has any questions so far.
Is one question from Ambrose. Is it fair to say that microbes are responsible for plant nutrition? What I remember from middle school is nutrients were created through photosynthesis.
That's a great question. So this question has been asked to Dr. White. Dr. White graced us with his presence at the last winter conference, and the same question was asked. And the question was, through this rhizophagy cycle, how much nutrition are the plants getting? And he gave a conservative estimate, saying, around 70% of the nutrition can be coming from this rhizophagy cycle, but also admitted that there could be more of this nutrition that they're actually getting from the microbial community. However, I also don't want to sell this story that, that microbes can do it all either. I think it's really a fantasy. I strongly believe that microbes are responsible for all of these processes, but we also have to remember, when in the context of agriculture, farming is not a natural thing, right? And because it's not a natural thing, we are a lot of times, these plants are putting a lot of energy into photosynthesis. They're putting a lot of energy into root exudation. A ton of energy goes into sending these root exudates to attract microbes. So it's part of their nature to do this, right? But in agriculture, we are somewhat extractive, right? We actually are taking some of these we're taking these plants away from where they grew. So all of those minerals that the plant extracted are now out of the soil, and we consume them. That's part of it. So we have to also strike a balance between that reality and also the fact that, you know, yes, they are actually microbes are providing a ton of nutrition, upwards up to 70%. Thank you for the question. Anything else? Okay, let's move on. Oopsie. I think I went too far. Or am I? Oh, sorry. No, actually. So I wanted to, speaking of, I wanted to bring a real case scenario. This is from a gentleman who went through the Soil Food Web School, and part of being a consultant, you have to have a final project. His final project, and this is Simeon from Hiawassee Products, he was able to, for his final project, he was able to create compost extracts. And for those who don't know, compost extracts are essentially taking a high quality compost, and by high quality, I mean it is what the Soil Food Web considers biocomplete, meaning that it actually has all of those trophic levels that we talked about present in there at certain concentrations. So he took this highly microbially rich compost and made an extract out of it. All you do is you're actually putting water through it and extracting those microbes off of the substrate that is of the compost. He then actually did a series of soil drenches on this plot, and they measured a few things. And if you want to see, I only took snippets of this. If you want to see more of that, you can actually go to Hiawassee Products and see more details of this. But some of the things that I wanted to bring up was, you know, what increased and some of the things that were a positive gain. Now, this was actually very short. You know, he did want to continue this process, but in this short amount of time, which I think was just 4 months, if anything, they were able to see gains. They were able to see gains in fungal biomass. They were able to see gains in protozoan biomass, which are our predators. They were able to test the plant sap and look at what the nutrition needs were. There were gains on that. There were actually lower nitrates too, which could be detrimental to plants. And they also found that the cation exchange capacity also increased. And this is a big deal because when we think about cation exchange capacity, we're looking at we're looking at the retention of some of these minerals. And let's take, for example, if this were somebody managing a golf course or any landscaping, and they were to apply this to their soil, and this will actually avoid some of the big issues that we're seeing in our rivers and streams and eventually the ocean, which is eutrophication. Eutrophication is essentially the addition of a high concentration of nutrients in waterways, which shouldn't belong in waterways, which are responsible for algal blooms and just a number of issues in our waterways. So this actually, this was a great experiment, one that could and should be replicated. Anybody managing lawns or even in your garden farming, this just goes to show you that there's a way that how chemistry actually has an effect on these systems. All right. So I was supposed to pause here, but we already took a question. So let's actually go ahead and continue with our tools of the trade here. I would say right now, this is not my favorite microscope image, but it's what I could find that from my arsenal of pictures. Anyways, eyepiece is actually not showing up here, but that is an eyepiece there. Down here, we actually have a mirror, but we actually don't use mirrors. We actually use light source. Nowadays, you're seeing more LEDs come through. And I just wanted to highlight only a few things in here of the microscope. And the reason why I want to highlight them is because, oh, and diaphragm is actually misspelled here. One of the things that I wanted to highlight these things here, and the reason for that is because the things that I have highlighted here are important for the work that we're going to be doing with these microbes. These microbes actually have the same refractive index as water, meaning that they, if you were to shine light through them, which we are doing with a microscope, you wouldn't see them unless you would shadow them, right? And so part of doing this work, in order for you to actually get a nice crisp image, there's a few things that are important. But let's start with the objective lens here. The objective lens, typically we actually have a magnification at the objective lens right here. We usually work at 4x, 10x, and 40x. 40x is where we do most of our work. And then the eyepiece actually adds another magnification there too, typically 10x. So you stack those, which is why it's a compound microscope. You have your compounding objective or magnifications, right? So you're compounding your eyepiece, you're compounding your objectives. And we're usually working around that 40x magnification is where we actually can see these things. And I'll show you some pictures of what that looks like ahead. Then we have our coarse focus. I've never seen a coarse focus appear. They're typically down here along with the fine. That's where you actually get your stage and everything set up there at your low magnification, so that when you're actually flipping these, you're not crushing your glass. Your fine focus then is actually, once you have that all dialed in, you're only using your fine focus after that. And then you have the iris diaphragm and your condenser. The iris diaphragm, just think about it as your eyes too, right? When you look at your eyes, your iris, there's that opening and closing. And all that's doing is actually allowing a certain amount of light to go through. That's important when you're actually looking through this, so that you're not blinding yourself and blasting your eyes with all that light that's coming from the light source. And also that allows, again, for that shadowing of these microorganisms, right? And then finally, you have your condenser. Your condenser is actually going to be here at the bottom as well, and that's going to allow a certain amount of light to come through. And where that comes into play is so that you can actually fix your background, so that your background is not marbled or, looks like the stars or something like that. So let's actually see what these organisms look like, right? These are some of the things that you may encounter when you're looking under the microscope. We already saw the nematode earlier, but I want to point you at the fungal hyphae and protists over here. As you can see, these are actually two testate amoeba. Testate meaning that they actually live in a shell and have like a pseudopod. Pseudopod meaning like an arm, if you will, that crawls out, and they do all their functions that way. And then here you actually have a fungal hyphae. This is probably of Basidiomycete based on the septa. You can see the septa here. There's just cross walls throughout. It's branching, it's nice and healthy, it's dark. There's theories that this melanin is actually coming from to protect themselves from solar radiation. And in all of these, you will also see bacteria. And this is actually something interesting, right? For when we're looking at the microscope, you're actually looking at a lot of layers of water. So even though you're actually putting one single drop there, you're seeing a lot of layers. And so when you're looking for bacteria, you have to look for something that's completely, first of all, you can see them at 4x, but in order to identify them and distinguish them from clay particles, you need to go into 400x magnification. And if you want to look at bacteria that are coxy, meaning round, they would be perfectly round and without edges or jagged pieces. Next, we actually have fungal spore and cyst. So this dark oval shaped particle here, it's a fungal spore. That's just one fungal spore. There are many, many different types, which you can actually use certain dyes or morphology to figure out what fungal body it belongs to. Again, here we actually have two protozoa again, similar to the one over here. So these are also testate amoeba. And the reason why I chose this picture was because there's a few things happening in here as well. You know, you have your microaggregates here, you have your sand particles here, or mineral particles. But here to the left side here, you're also seeing what's called a cyst. Now there's a double wall on this, and there's all this nice stuff in the inside is active, or not active, but it's alive. And that's actually an amoeba cyst. So all it's doing is it's one of these guys that actually has cysted up. And what they do is, the reason why they do that is because the conditions are not right for them. So once the conditions are right, they will actually come right out of that dormant stage and start functioning as well. And lastly, just so that you can see a little bit of bacterial diversity, this strand right here, it's actually a actinobacteria. So that just goes to show you the type of morphology that we follow in terms of bacteria. Down here, I'm not going to go into much detail here. This is actually a nematode. I believe it's a fungal feeding nematode. Typically, you could see a small stylet here, which indicates that they actually feed on fungi. And there's fungal hyphae here as well. And lastly, you actually have a columbella, also known as springtails. These are actually decomposers that are needed for, part of this whole nutrient cycling piece. And that is the actual color. This is not an effect. They range from gray all the way to purple. And they are excellent to have around. So if you do see them around, know that this is actually a good soil health indicator, along with earthworms, right? As we saw in the diagram of the soil food web. So the next tool that I want to get into is the microBiometer. The microBiometer is or was adopted or created by a soil scientist that understood the need for quick, easy, and cheap evaluations of microbes in the soil. And like with the microscope, I have selected a few highlights of what you can actually find in this. First, there's a whisker in here. The whisker actually breaks up all of those, and this is the whisker right in here. Once you collect your sample, it breaks up all of those aggregates to make sure that the microbes come out of those aggregates and into solution. And then there's a reagent. I haven't been told what's in there, but I assume that there's actually salt and perhaps a flocculant as well. Salt helps out, what they call in organic chemistry, salt out the organics. And in addition to that, the flocculant allows for the precipitate to actually fall, and everything else remains in suspension. So that when you take your sample, you're getting what you need out of there. There's a soil sifter. The soil sifter is actually there to maintain a homogeneous collection of these samples. And finally, the test card, which is here. It uses, and actually we can go to the next slide. So it uses this color gradient here. It uses grayscale. This is something that you actually, it's an app on your phone, and this is a screenshot of the much larger things that you get from here. But in general, when you're looking at the soil sample results, unlike the microscope, you're not getting to understand what the diversity looks like, but you're getting to see the total microbial population, right? In addition to this total microbial population, you're also getting fungi and bacteria counts so that you can see this fungal to bacteria ratio. There's many people that believe that these fungal to bacteria ratios correspond to plant succession, meaning that if you actually have bare soils, your fungal to bacteria ratio is low, and on the other end of that spectrum, in succession, like let's say an old growth forest, you would have a high fungal to bacteria ratio. So it provides you those levels there too, along with, again, the total microbial biomass. The folks at microBiometer have also established some metrics here so that when you're utilizing this this app, you can have something to guide you with. This is excellent for evaluating change over time, evaluating your practices. So if you actually have something that you're trying out, either management or a new product, this is a good way to know, okay, well, did this thing work? Or am I heading in the right direction? Or am I actually causing detrimental effects to my microbial population by adding a certain amendment at a certain time of the year? So great tool to have. It's easily accessible and at the reach of many. And before we continue, I want to take another quick pause here. There's a quick, and for those that actually arrived earlier, I was already asking this question, but I would like to record this question. And the question is, it's more on assessments needs. So there's about, I believe, 2 questions on the needs of, or tool needs, if you will. So if you can, if I didn't bore everybody to death and everybody's asleep, you can actually try to answer as best as you can. And there are 2 questions there. All right, I think we can close this. We have 87%. No, wait. 90%. All right. Yep. All right, let's give it to 115. All right, cool. All right, wonderful. It seems like people are interested in both of these tools, and this is great information for us to have. This helps us, this informs us as to, the needs of our membership and abroad too. Not just memberships, but also anybody that follows NOFAMAS, anybody that's concerned with the environment, anybody that's concerned with farming, growing anything, any eaters, producers, basically the general population should be, invested in this. So thank you for that. That again will help us determine programming in the future as well. And I guess before we continue, any questions so far? I think.
Ruben, I have one.
Yes.
This is Juan Carlos from Sobremesa Farm. Have you considered using chromatography as a tool too for these kind of tests?
Juan Carlos, thank you so much for that question. I have been, I went through La Mierda de Vaca online courses, and I am a firm believer that that is certainly a helpful, an extremely helpful tool, especially for the you know, paper chromatography takes that, brings that sovereignty back to the producer. This is something that anybody can learn, and it's not difficult. And in fact, I will be doing an intro class in the spring or in the fall, specifically on a section of paper chromatography, which I'm more interested in, and that's the humic acid, fulvic acid, and hematomic acid fractionation to quantify humification in the soil. That is certainly something that it's dear to my heart, and it's certainly a great way to evaluate soil health in general, but also tracking these this humification in the soil as well. But and as you know, paper chromatography, for those that don't know, paper chromatography is a way to evaluate many things in the soil. One of them is soil health in general. It shows you how the mineral profile is connected to the microbial profile, to this humification profile as well, and how these things are meshed. You can also look at contamination through this. So you can also evaluate it that way. Yes, Juan Carlos, this is certainly something that we will be pushing forward as well. And I'm hoping to have more conversations with you about Haido coming to the US again. So hopefully that's something that we can actually collaborate as well. So yeah, thank you for that question.
Absolutely. Sounds good. Thank you.
All right. Oopsie. So I'm going to stop my share here now. Oh, wow. I didn't realize I was so dark in here. All right. So, I think we can open this up for any other questions, anything else that, any reflections that we may have. Are people overwhelmed with this information? Does it seem attainable? Is NoFaMAS going crazy? You know, did we go off the rails and, you know, we're thinking some crazy thoughts here? Anything that comes to mind?
I will say, I'm Brianna Alfaro. I work with Soulfire Farm. Like I wanted to say earlier in the chat, but you said that you didn't have access to the chat, that this is not boring at all. I'm not falling asleep. I'm very fascinated by this, and I had considered participating in the, you know, the soil food web and like bigger curriculum. But I just couldn't commit to it this year. And so this really felt like a right size introductory opportunity for me, this whole series. So I'm really grateful. And coming from like a privileged perspective of like, I do have a budget for a microscope, I would be interested if you have like a resource to share on like things to consider when like shopping for a microscope for soil analysis.
I will, first of all, thank you very much for that, those for your kind words. It's good to know that we're not that crazy. But that's an excellent question, and this is not scripted, by the way. This this question was not scripted, but I will take the opportunity to say that our board member John Duke, which is actually in this call right now, has been working with a company called LW Scientific. LW Scientific provides laboratory products, and we worked, or John actually led this, so that NOFA members do get a 40, 30, 35, 37% discount on all products on their website. And you could become a NOFA member for as little as $10. So this is a great plug for our membership, but also we realize also that this is going to help the community, as you all were able to see. There seems to be a need for having these tools to evaluate the microbiology in the soil, and if we can actually help the community gain some of these tools, we're all about that. So as far as your question, maybe that's something that is a question that we often get, and maybe that's something that we can actually put up on our website. But if you can email me, I'm right on the website. You can email me and I can send you specifics. Or after this, I wanted to send you all a list of resources. So in that list of resources, I will include, microscope specifications too.
Thanks so much.
Thank you. Thank you for the question. Had you not had to ask that question, I probably would have forgotten to actually mention that. So I appreciate that. All right. So yes, thank you, John. Yeah, we on that note too, we are also offering a course, a microscopy course with our partner sister organization, Connecticut NOFA. And I don't know if somebody can help me actually find that and paste it on the chat. That's a course that's actually being actively offered. That's going to start up in April, actually. There's going to be different locations throughout Massachusetts and Connecticut. And then there's going to be an online component as well. So if interested, that's actually an opportunity also that we're offering. There's more details on that. It's $600, but you actually do end up with a microscope. So think about it as a $300 course. Thank you, guys. Let's see. There's some questions here. I think I saw one here. Can you speak about mycorrhizae and amending soil? Yes. So we can speak about mycorrhizae, but is there something in particular that you would be interested in mycorrhizae? Mycorrhizae do form plant associations. They essentially go in and infect the roots, and there's different types of mycorrhizae. There's endo and ecto. Endo meaning that they infect the inside, ecto the outside, and they are certainly highly beneficial. However, they are sometimes inhibited by high levels of phosphorus in the soil. And as Laura Davis, that's here, can tell you, most of the soil samples that we get in agricultural soils have very, very high phosphorus. Now, there's ways to cultivate your mycorrhizae, and then there's also microbial products that you can purchase. Now, a question for you is, how do you know that your roots were inoculated when you added your microbial inoculum, right? You know, because we think, okay, well, there's a lot of times when you buy them, there's a lot of species in there. Did that species work with that particular plant? How do you know that worked? And that's a rhetorical question because I have an answer for you. There's a couple different ways you can do this. There's a very there's a on-farm way to stain these roots, which involves ink and some replacement chemicals so that they're not too harsh, such as glycol and acetic acid, vinegar. So there's ways to do it that way. An easier way, but actually not always accessible, is if you actually had an attachment to your microscope. So when we talked about the microscope, we only were talking about light microscopy, right? But there's other attachments that you can actually add to specific microscopes, and in that case would be called epifluorescence. So with epifluorescence, you can actually, let's say you have a mycorrhizal inoculum. You added it to your soil. You're like, okay, well, did that work this year, or did that work on that specific plant? You can dig in and take a little piece of that root and put it directly under the microscope with the epifluorescence, and it will show you whether or not your root was colonized, and at what percentage as well, because it will autofluoresce with this light. So that's actually, you know, another aspect of microscopy that you can be using, not only to evaluate your microbial population, but, you know, did that mycorrhizal inoculum work? Anything else specifically to mycorrhizae? Okay. Ambrose, she had a question on synthetic fertilizer interactions with soil biology. That is great. So in speaking with Dr. White, he has brought us a lot of insight on this because, you know, I think for the most part, everybody knew that synthetic fertilizers were bad, right? We just knew inherently that they're just bad. But one thing that he has, you know, brought science into this in a very important aspect is that we now know, and this is where, you know, I have to be careful to how I say this, because not just synthetic fertilizers, but fertilizers in general can have detrimental effects on soil microbes. For example, and I'm not saying don't do it. I'm just saying, you know, this is something that we should be that we should know. Say if you're actually adding any nitrogen sources to your soil, you have free living bacteria that fix nitrogen, and you also have bacteria that are symbiotic to plants. Once you start adding this, those numbers actually decline because now the plant is saying, well, no, I'm getting all of that, everything I need, this human is giving it to me. So I don't need to actually interact with these microbes anymore. So this is it's a cautionary tale, because again, these plants have evolved to obtain their nutrition from these microbes. So not just synthetic fertilizers, but even more so, some of the work that Dr. White is doing has cautioned us against some of these other chemicals that the plants have forgotten how to interact with microbes now. And that is very, very problematic, because when we're talking about seed breeding, you know, genetics, this is something that should concern us all, right? You know, where are we getting our seeds from? This goes back to the importance of all of these organizations trying to do the right thing with, you know, saving seeds, promoting seed saving. This actually goes back to that whole rhizophagy cycle that we were talking about earlier. and why it's important to actually promote these practices. So yeah, not only that, there's also the the obvious problem with salt buildup in your soils when you're using synthetic fertilizers too. So yeah, thanks for that question. There's and I would like to quote Dan Kittredge actually on this. Dan Kittredge, I remember at our last summer conference, he mentioned, he said something to the effect of, just because your plants are green doesn't make them healthy. And a lot of times when we're using these synthetic fertilizers, yes, these plants look nice and healthy and green and lush. But have they interacted with these microbes? Do they actually have the right nutrition? So these are some of the questions that we should all be asking ourselves. And I encourage you all, if you haven't actually been to the Bionutrient Food Association website, please visit it. Dan Kittredge is really at the forefront of defining nutrition, and I think that it's a paradigm shift that we should all be thinking about as how we define this nutrition. And the reason why I bring it up is because it transcends any soil practice, any certification. At the end of the day, am I producing healthy plants? Because these healthy plants will give us healthy foods, which is what we need for our brains to properly develop and have beautiful conversations like these, right? So I'll get off my monologue there. Let's see, any other questions here? We will have access to the recording. Yes, this will go onto our website. For those who don't know, we actually, NOFA has a YouTube channel, and a lot of content. There's, as you all know, there's been a lot of conferences throughout the years. After 6 months, those conferences recordings actually get uploaded there, all these webinars as well. So there's a wealth of information on that YouTube channel. So I encourage you all to actually try to check that out. So what happens to the soil if the plants do not interact with microbes? We enter this perpetual loop of this problem that we have, right? So if the plants don't know how to get to that nutrition, we're going to have to basically give them an IV, right? Because they don't know how to get it there themselves. So now we actually give them this IV. They're on the drip now. And the only way they can feed themselves is through, diffusion, through this process. Now, the problem with that is if they don't know how to interact with microbes anymore, hopefully I made this clear, that these microbes are also responsible for protecting these plants. So if they don't have this first line of defense, this means that, we gotta now spray it with this pesticide. Now we gotta spray it with this other stuff. So then we enter this perpetual loop that we can't get out of because they have ceased to actually have their natural functions. And that's problematic because that, I want to quote Chief Seattle here, and I'm paraphrasing here, but over 100 years ago, he warned us about this, that everything that we do has an effect on, anything that humans do has an effect on us all. Nothing is done on a silo. Everything that we do has an effect on the web. And we didn't heed the warning. And here we are now. But that all said, I think I have high hopes. I think that information like this is being disseminated widely. People after the pandemic have learned that, where's my food coming from and the importance of food. So I'm hoping that we can continue riding that wave and hopefully demand for these things. So thank you for that question. Compost tea is adding microbes without adding organic matter. Is the benefit of compost tea merely short term, or are their benefits perhaps overhyped? Compost tea is an excellent way to get biology into the soil. I think that the problem with the bad name that compost tea gets is the fact that people didn't have microscopes to make sure that that their compost was highly microbially active to begin with, right? So if you actually are doing a compost tea, and for those that don't know, compost tea is you add compost to a system where you aerate it for some amount of time. Sometimes you add food, sometimes you don't, but you're aerating that compost and you're using the liquid portion of that to spray on your plants. That is a compost tea, which brews for a certain amount of time. So I think that the bad rep that it gets, again, it's because people didn't have a way to evaluate how microbially active was that compost to begin with, right? There's also the argument about compost teas versus extracts, that extracts provide you with and I've seen the data, that compost extracts are more diverse, and the compost teas are less diverse, which makes sense, right? You're actually, when you're doing a compost extract, you're taking everything without changing that compost. But when you're doing a tea, you are selecting for microbes that want to live in that highly aerated environment. With specific foods that you're giving it. However, that said, you can control these, you can control the air, you can control the foods. And if you actually have a pretty diverse compost to begin with, compost teas can be an excellent way to actually increase your microbial populations. They inhibit pathogens, especially if you're spraying that onto plants. They are also part of nutrient cycling on the plant as well. This is something that we actually learned from Dr. White as well, that plants are not only feeding through their roots, they're feeding through their leaves as well. And if we know now what we know about rhizophagy, if you're spraying these things right onto them, they will actually internalize them as well, and fix nitrogen or other nutrients too. And they do this a lot through either the stomata or the trichomes on the plants. But yeah, no, I wouldn't say that they're overhyped at all. It's just, I think it's just people need a way to actually make sure that they're doing the right thing. Hope that answered the question. Yeah, and that's actually just to follow up on that too, whether or not it's actually a short term, there's that is an argument to be had, whether or not it's short term or not. But again, one of the things that do happen when you're brewing and you're brewing right, you can actually feel that stickiness starts to form. Those are the microbial glues happening. So if you spray that onto your plant, now you actually have protection that lasts a little longer because it actually has some of these microbial glues that are protecting the plant from any potential pathogens, and also it doesn't just get washed away too. So it should have, at least for our annual plants, enough protection. Yeah, so thanks, John. There may not be organic matter, but there's a humic aspect for sure. And I want to add to that too, not only just the humic component to that, but there's also chemical signals, right? So we now know about these things called autoinducers. Autoinducers are hormone-like, and fulvic too, yes, hormone-like chemicals that basically are the communications for these microbes. So, so say if I actually have and this, by the way, actually is the same as microbes in our gut, microbes everywhere. This is just the way they communicate. Say I actually have a microbe that is sending out a signal to say, hey, I'm here. Where are my cousins at? And they say, okay, where are my cousins over here? So there's a autoinducer for that. Okay, well, where are the ones that are just like me? Okay, there's an autoinducer for that. Are there any predators in the system? Let me put an autoinducer for that. Are there any you know, is there any competition? Anything else that's not like me? And so when you're putting these compost teas out, you're also putting all these chemical signals out into the world. And those will also either help or deter, depending on who was in that compost. Um, uh, spread these autoinducers and send these chemical signals and say, hey, let's say Fusarium, don't come over here because you're not invited. There's so many of us over here, and this party's not for you. So there's many aspects of compost teas that are, I guess not well known. So does bokashi have a different effect on plants than warm compost tea? Again, these are actually, bokashi is, to clarify, bokashi is a Japanese way of fermenting, and it relies on a group of 3 microbes, yeast, lactic acid bacteria, and photosynthetic bacteria. These are considered effective microbes. So that's what bokashi is. You have these 3 specific microbes, and that's going to differ very, very vastly from warm castings, right? It's going to be a complete different population. So you're going to actually have complete different signaling. Going back to these chemical signals, you're going to have complete different signals that are in your bokashi versus your warm tea. So, because there's different signaling, you're definitely going to have different effects on your plants. So, and one of the things too that I would argue too is that sometimes some of these extracts or teas are better suited for one thing or another. For example, the extracts are better to be used as a soil drench. The teas are actually more of a foliar. Warm, Johnson Sioux composting, which is a specific type of composting, is probably better suited for seed treatment as a seed inoculum. So there's, one thing is not better than the other. I think that there's just, different applications for this. And understanding some of these basic concepts of, who's in there, who's into your bokashi, who's in your compost tea, who's in your warm tea, will help you understand, how that can actually help with your plant. These are great questions. Thank you. Anything else we have here?
Hey, Ruben, I have a
Christine.
question.
Hey.
So I have a question. So I've used microscopes extensively. I have access to one, but I've never used it to look at the soil. How hard would it be for me to scoop some soil and actually look and actually find some of these microbes, assuming that they actually are in my soil and they haven't been totally killed from being mismanaged for forever?
Yeah, so it's we have actually done like 1 day one day workshops or and with different groups, all the way from high school to college and in between. And in that very brief amount of time, everybody actually gets it. It's super simple. All you're doing is you're taking some of your substrate, whether that's compost, whether that's soil, you're diluting it in water, shaking it up a little bit, letting it settle for a little bit, and then you're taking that supernatant water, just water from the top, avoiding the organic material, putting it onto a drop. Will all microbes show up there? Some microbes are a little easier than others to come off of their substrate. But for the most part, it's a simple process, just shaking them up a little bit without killing them, and then putting on a slide, and there you have it. There's other methods too that are specific to extracting certain microbes. So like, for example, there's like microbe traps that you put out for, let's say, mites or other arthropods. There's also ways to extract nematodes as well, that are slightly different from that, but the process is still the same. So it's I would say it's a very easy process. And again, my 4-year-old is allowed to do some things on my microscope. But she's, she now knows, okay, well, I know that there's water bears in this moss, and she'll sometimes come home and like, oh, can we look at this water bear in this moss? And and I'll let her move the microscope around and navigate that. And so it's an easy process, Um, and I want to, and thank you for that question too, because I want to demystify the fact that, that microbes belong only in labs or the classroom or, in academia. These instruments are very, very easy to use, and nobody should feel intimidated. When you start looking at these microbes, it's you'll easily start to identify them, and for the most part, we're not speciating anything either, right? We're not saying, oh, this is E. coli. We don't know what it is. We're only looking at morphology, right? We're only looking, is it a rectangle? Is it oval? Is it circular? For bacteria, for fungi, how long is it? How thick is it? Does it have walls? For the protozoa, a lot of times we can't identify them because they're moving too fast. Well, actually, that one that moved too fast is a cilia because of the way it's moving, right? Or it's a flagellate because it's bumbling around. So it's only a handful of things that we're looking at as well. When you looked at that soil food web chart, that's, it's only a few things that we're looking at. So the more and more you do it, the easier it becomes. But yeah, thanks for that question.
Thanks.
I think that we were supposed to go up to 7:30. I don't even know. I just work here. But yeah, so I also want to put in a plug for a few things that are happening coming up. But please visit our website. We have a ton of events coming up this year. I think we have over 20 events that we are doing this year, and I'm very, very excited for all these events. We have a beginner farmer. Oh, thank you, Christine. We have a beginner farmer series that we will be putting out dates in a few days, hopefully. Those are going to be held at Farming is Life in Winchendon and Woven Roots in Tyringham. Each one of those will actually be a 3-part series focusing on the 3 pillars of soil health, one of them being the chemical, physical, and biological. We also have a compost tea workshop that's up on the website now. That's happening at the end of April, I believe April 27th. That's going to be in Metapoiset. So if anybody wants to learn more about compost tea, that'll be an excellent workshop there. We will be diving deep into some of these questions that arose that showed up today in this meeting. We also have, again, back to the beginning of this call, this series. This is the kickoff to the series where this was just to give people a basic understanding of, why are we doing this? Why is it important for these microbes in terms of agriculture, and what are the tools that we're using to evaluate them? Part of what we want to try to achieve here at the end of the year is, sign up, go to 1, go to 2, bring your soil. We will look at the soil under the microscope. We will do the microBiometer as well. And then we'll just talk about these things. With that, also a survey accompanies your soil too, so that we know what soil type you have, what type of management you're using. The purpose of all this is that at the end of this series, we would actually have a better understanding. I'm not going to say we're going to have a complete understanding, but we'll have a better understanding of, how does management correlate to microbial communities. And if we can actually at least have 2 things that come out of that, and we can broadcast it to the entire state and beyond, I think that we would be a little better in terms of understanding. And I know for a fact, because I recently spoke with a farmer that when I told him about this, expressed how important it is for us to do this, because they don't have the time to do this. But it'll advance all our knowledge on this aspect. So I hope that you can join. These will be in June. I believe it's June 14th or something like that. Anyways, with more information on the website. And in August. I don't think we have determined the date in August, but we'll have those 2. And hopefully, by the end of that, we will have a roundtable at our winter conference to just so that everybody can actually benefit from that. And even in 6 months after that, the recordings will be made available as well. So I think that's it. Gave a plug for our membership at events. Yeah, we're moving fast, and I'm hoping that you all can come along with us as well. This is exciting stuff. So anything else before we leave? Anybody has any? I don't have any closing remarks. Just come with us. It's fun.
Ruben, there was an additional question from Melissa about soil probes.
Oh, wait, what?
Melissa Curtin asked, do you have a particular soil probe route you'd recommend?
Soil probe for taking samples? Is it for this particular? So Dr. Elaine Ingham actually says, "Oh, just go ahead and use a soil or an apple core." And that's the person that's the leading scientist in all this too. So she's like, just use an apple core. Again, there's nothing complicated about this. And I truly believe in citizen science as well. And even Dr. White has mentioned this too. He's like, we need more people doing this. I have the academia, I can do this in the lab, but until it goes out into the real world and we all are doing this and doing our part, it means nothing really, because again, it's a lab scenario, and we need more people doing this work. So yeah, just an apple core.
Okay.
Wonderful.
Anything else?
Oh, so I thought that the chemical composition of the probe might affect samples. Not necessarily. I wouldn't worry about it unless it's like copper or something. But, well, thank you so much. Thank you all for joining us in this little adventure here. And I hope to see some of you soon. And you all take care.
Thanks a lot, Ruben.
Thank you.